Hi. So today we are going to discuss the third session of our chapter Bioavailability and Bioequivalence. The last two sessions we discussed regarding the bioavailability as well as the dissolution criteria, etc. So you know what is bioavailability, determination of bioavailability, and dissolution, what are the dissolution apparatus, acceptance criteria, etc. Okay. Now in vitro, in vivo correlation and bioequivalence. These are the two things what we are going to learn in this session. In vitro, in vivo correlation as well as bioequivalence. So first of all, let me tell you what is in vitro, in vivo correlation. This is, if you read the textbook, you may feel some difficulty to understand the concept. So I wish you just listen what I am saying now. Okay. This is purely a relationship between the in vitro behavior of a drug and an in vivo behavior of a drug. Means we are developing a relationship between in vitro dissolution and in vivo bioavailability. The last session we studied dissolution is an in vitro method for determination of bioavailability. So this in vitro method and in vivo method, bringing a correlation or developing a correlation, developing a relationship between the in vitro test and in vivo test. What is the necessity for this? Now, this IV-IV correlation serves as an alternative to avoid various in vivo studies. As you know, the conductance of in vivo studies is very costly, tedious. It includes many manpower, number of volunteers. It's very huge. And getting the ethical clearance, ethical considerations, patient health, welfareness, everything must be considered before going for a clinical study. So the purpose of IVIVC is to avoid the clinical study and compare the in vitro test with the already available in vivo data. Compare the in vitro results with the already available data. This is the concept behind IVIVC. So the main purpose is to reduce the cost of developing and considering the ethical importance, avoid the clinical studies. Now, how to build a relationship? I'll give you an overall idea first of all, then we'll go through in deep into the sessions. Okay, suppose we have a particular dosage form, or an innovator, the product, the person, the company who has invented the product, they invented it, they patent the product. Now I'm going to develop a generic product of it. Consider in this way. So you are an innovator product, and I am going to generate a generic product for that. You being an innovator person has to do all the clinical trials, means preclinical, phase 1, 2, 3, etc. before launching the drug into the product. You know very well what are the various phases. Now, for a generic product, for a generic industry to launch the same product into the market after the patent duration of the innovator product, the generic company or generic industry need not to conduct all the clinical trials of 0, 1, 2, 3 in order to launch the drug product into the market. Rather, he has to prove only the bioequivalence of the product. This is only in the case if it is a generic product. a manufacturer or a company is developing a generic product. So considering the innovator, they invented the product, they done all the preclinical trials, their cost is very huge. Where I am a generic company, I am just developing the product and I am doing a bioequivalence study which proves my product is equal to the innovator product. Bioequivalence in biology, in body, my product behaves same as that of the innovator product. My product reaches in the same rate that the innovator product same reaches. So a generic manufacturer always claim my product is equal to that of a innovator product. This is a reason the generic companies can produce the medicine at a very cheaper cost. The cost factor behind the innovator product is highly expensive for conducting the clinical trials, and the safety aspects is also very important. So in this way, the generic company can reduce the cost very much. Now they need to perform only the bioequivalence study. Once they developed the final product, only need to conduct the bioequivalence study. Suppose I am developing a formulation, I may have to do thousands of trials in order to end up with an optimized formula. So these 1000 trials I cannot perform bioequivalence study. The last optimized formula only I may go for the bioequivalence study. So my aim is to develop a product same as that of the innovator product. My aim is to develop a product same as that of the innovator product in all the characteristics. So here I'm doing many trials, I'm developing a product, and I am conducting or evaluating the last product only for the bioequivalence, not for any other clinical trials. Bioequivalence, conducting the bioequivalence is not a tedious or it's not a heavy task. Okay, in the other way it includes all the ethical considerations, but it's not a tedious one, comparing with that of the normal clinical trials. Now I made thousands of trials to optimize a formula. What strategy I can use to optimize the formula? How do I know this product? Because I'm doing only the in vitro study. I'm not doing the in vivo study. Bioequivalence is an in vivo study. Clinical trial is also in vivo study. In the laboratory condition, I am performing only the in vitro study, not in vivo study. So with the in vitro result, I should satisfy or I should have a confidence that this product will give a good result in vivo. How I can develop that confidence for my product to get a good result in vivo? That is done by the IVIVC. So before taking a drug product in for a bioequivalence study, the generic company or an innovator company also can compare their product with an IVIVC. Now what the generic company will do, so they will purchase the drug product from an innovator company. Okay, they will purchase a drug product from the innovator company. They will perform the in vitro studies in their laboratory conditions. And from the FDA or from the patent office, everywhere, they can get the in vivo details of the drug product. What is the rate of absorption? What is the Cmax? What is the AUC? What is the total bioavailability of the drug product? So and so, all the kinetic parameters, they can get it from the FDA website. Now they are performing the in vitro dissolution of the innovator product. So the way how you developed a standard graph, here you can develop a relationship between the in vitro dissolution and in vivo plasma drug concentration. A standard relationship between in vitro dissolution and in vivo plasma concentration, or in vivo Cmax or AUC, whatever it might be. For example, you consider we are developing a standard relationship between the plasma concentration and in vitro dissolution. So you make a graph. In the y-axis as plasma concentration, x-axis as in vitro dissolution. This is a very simple example. Okay, in the x-axis in vitro dissolution and in y-axis in vivo plasma concentration. 10th minute, you done the dissolution study for 1 hour. You collected the plasma concentration data for 1 hour from the data from the FDA website. So in vitro, at the 10th minute, it produces a particular percentage of drug release, whereas in the 10th minute, what is the amount of drug reached in vivo? So by plotting a graph, a standard relationship you can build between the in vitro dissolution and in vivo plasma concentration. So this relationship is for what? This relationship is for the innovator product, or the product which we are considering as a standard. So if we develop a standard relationship between the in vitro and in vivo performance, this relationship we call it as the in vitro in vivo correlation, a relationship between the in vitro and in vitro behavior of the drug with the in vivo behavior of the drug. I hope now we got an idea what is an in vitro in vivo correlation. Now we'll go through the sessions. A predictive mathematical model that describes the relationship between an in vitro property of the dosage form and an in vivo response. Hope you are clear. In establishing a relationship usually linear between the in vitro dissolution and in vivo bioavailability parameters, that your kinetic parameters, by using data from previous bioavailability studies to modify the dissolution methodology. So previous bioavailability studies means what is available in the FDA website or is a published one that you can take as a bioavailability study. So what is the advantage of the generic company? Their cost is they are taking the data of the previously published one in the FDA website or published articles, bioavailability data. So no need to spend money for conducting the bioavailability study. They need to purchase the innovator product and do the dissolution in their laboratory condition that they have to perform. That is the only investment for a generic company to make an in vitro in vivo relationship. Now what they will do, they got a relationship. Now the generic company, they can manufacture the dosage form, and this dosage form they can compare with the standard one. Means suppose the standard drug product in the 10th minute they got 15% dissolution. Your drug product also you should get 10th minute 15% dissolution in order to get the in vivo behavior for the same in vivo behavior to that of the standard drug product. I'll repeat. If the standard product has an in vitro dissolution of 15% in 10th minute, you can set your goal for your product as 15% for 10th minute. Then and there only you can achieve the same in vivo behavior. So this way you can develop the product. So in vivo correlation can be correlation based on the plasma level data, correlation based on urinary excretion data, correlation based on pharmacological response method. Same how you perform the bioavailability studies. You can consider the kinetic parameters, plasma kinetic parameters like Cmax, Tmax or AUC, whatever it is. Urinary parameters like XU, TUX or TU max. So those type of urinary excretion data parameters can be used. Moreover, the response method also, pharmacological response method also can be measured. So these three things can be used. The three data can be used to develop an in vitro in vivo correlation. I'll come back to this. So there are various levels of correlation: level A, level B, level C, and multiple level C correlation. This is level A correlation. If you look, the x-axis is drug dissolved. The graph, look into the graph. X-axis is the drug dissolved, y-axis is the drug absorbed. This is the highest category of correlation, which represents a point-to-point correlation. That means in every point, the 10th, if percentage of drug dissolved, 20% it dissolves, this much percentage is reached into the systemic circulation. When it dissolves in vivo 40%, it reaches this much percentage in the systemic circulation. When it dissolves 80%, this much percentage has reached into the systemic circulation. So each time point, what is the drug dissolved in vitro is compared with each time point what is the amount of drug reached in vivo. Point to point correlation. This is the level A correlation. Okay, so we are developing the correlation and we are comparing that with our test product with the standard product. So this is the level A correlation. Coming to level B correlation. So a minor difference is always there. Mean dissolution time in vitro is compared with the mean residence time in vivo. This is not a point-to-point correlation. Mean dissolution time and mean residence time. Mean residence time is a non-compartmental calculation method that I'll teach you when we are going through the non-compartmental analysis. Mean residence time, AUC, AMC and all, have to determine the mean residence time. Mean residence time generally says how much time the drug resides in our body. Okay, and mean dissolution time. So mean dissolution time is combined with mean residence time. In the previous level A, we have each point we have compared. Here each parameter is compared. Any two parameters, one in vitro parameter compared with another in vivo parameter. So this is not a point-to-point correlation and does not reflect the actual in vivo plasma level curves. So in the previous one, exactly we are comparing the plasma concentrations. Okay, here we are considering only the mean residence time. This this method has its own drawbacks. Next one is level C correlation. This is also a single point correlation, same as that of level B, but other than the mean residence time of mean dissolution time, some other critical kinetic parameters are taken into consideration here. That is T50, dissolution T50, T90 means the time taken for the drug to dissolve 50% is the T50. T90 is the time taken for 90% of the drug to dissolve, is combined with the one mean kinetic parameter Cmax or Tmax or AUC. The relationship is between the plasma concentration time kinetic parameters Cmax, Tmax, AUC with the T50 or T90. So this is also a single point correlation, but directly we are comparing the kinetic parameters. In the level B, we are comparing the mean residence time and mean dissolution time. Okay, so this is the level C. And we have another level, that is the multiple level C correlation. It is a relationship between one or several pharmacokinetic parameters of interest and the amount of drug dissolved at several time point of dissolution. So this is not a single point comparison, but multiple parameters has been taken into consideration. For example, Cmax you can compare with the in vitro behavior. Tmax, for the same data you can compare Tmax with in vitro behavior. AUC can be taken into consideration, and each plasma drug concentration can be taken into consideration for in vivo, in vitro dissolution. So multiple kinetic parameters can be compared in the level C, where in case of level B, what was the difference? It's a single point comparison. T50 is compared with the Tmax. T90 is compared with the Cmax. So this is a single point comparison. Only one factor is taken into consideration, whereas in multiple level C correlation, any number of factors can be taken. Multiple number of factors can be taken into consideration for comparing or developing the standard behavior or comparing the relationship between in vitro and in vivo performance. Okay, so these are the various levels of correlation. Level A, B, C and C, multiple level correlation, and the correlation can be built for plasma, urine as well as pharmacological response method. Okay, what are the parameters for correlation? This is exactly same what you have learned in the plasma data. Means AUC and Cmax can be a time for specific amount of drug to dissolve is combined with the AUC as Cmax. That means level B correlation 50%. 50% time taken for 50% drug to dissolve is combined with AUC. Amount of drug dissolved at a time period, 10th minute, how much is the percentage dissolution is combined with what? Fraction absorbed, F or Ka, rate of absorption. Mean dissolution time is combined with the mean residence time. Mean dissolution is an in vitro parameter, mean residence time is an in vivo parameter. Parameter estimated after modeling the dissolution process, that is concentration at a time t and absorbed at a time t, that is in vitro concentration and in vivo concentration at a particular time point. So these are some of the parameters for correlation. Now this is all about the in vitro in vivo correlation. Hope you got an idea what is in vitro in vivo correlation. Now what happens now? A generic company who are developing the product who made a standard relationship, in vitro in vivo relationship first. With this relationship, they compared the product. They made a single formula or 2 or 3 formula. They made it. 2 or 3 formulations they made, and this formulation they want to compare with the standard one. So this comparison, in order to avoid the clinical trials, they can perform the bioequivalence study. Equivalence. So here we are going to identify what is equivalence, what is chemical equivalence, pharmaceutical equivalence, bioequivalence and therapeutic equivalence. Equal means what? Same. That's it. Okay. So one is biological equivalent, other one is chemically equivalent, other one is pharmaceutically equivalent, other one is therapeutically equivalent. That's all. It's not a major difference in this equivalent. So equivalent means 2 products are same. That's all. In the chemical equivalent means what? 2 drug products are having the same drug constituent in the active pharmaceutical constituent at the same amount. That's it. So same drug molecule, same amount, 50 mg metformin in the test, 50 mg metformin in the standard. So if both are assay in value, if both are same, purity if it is same, we can say they are chemically equivalent. So conducting the assay and purity of the test combined with that of the standard, identify both are of 50 mg metformin, purity is 99%, fine, it is chemically equivalent. What is pharmaceutically equivalent means what? So 2 drug products, they are identical in strength, quality, purity, content uniformity, disintegration, dissolution. Okay, so pharmaceutical characteristics must be same. Then and there only we can say they are pharmaceutically equivalent. Metformin 50 mg is the dose of the drug. Test standard generic, it should be 50 mg. The weight of the tablet is 100 mg for that both test and standard. The shape of the tablet, the color of the tablet. So the physical appearance of the tablet must be same. This you might have noticed. What is the generic company's size? Even the color of the primary packing material also will be same in case of the generic product. That means look same as that of the innovator product. Only the name will be different, or the brand name, the generic company's brand name, or the maybe named as generic name itself. It may be. Rest all the things look same. So they are pharmaceutically equivalent in quality, and in moreover. The last part is quality control test wise also. All the quality control tests, weight variation, hardness, thickness, friability, disintegration, dissolution, all the quality control tests also must be same as that of the standard. Then we call the product as pharmaceutically equivalent. Bioequivalence. The term itself, biologically, the concentration of drug that reached into the systemic circulation must be same as that of the test and standard. Means for the standard at the 10th minute 15% reached, means for the test also at 10th minute 15% of drug must reach. The Q value will be same always. At the 60th minute 100% of drug for standard has reached into the systemic circulation. Here also it should have same. So biological in vivo performance, in vivo kinetic parameters must be same for test and standard. In vivo kinetic parameters, what the body does to the drug. Okay, so Ka, Ke means moreover the rate of absorption, AUC, Cmax, Tmax, all these kinetic parameters must be equal. We call it as what? Bioequivalent. Therapeutic equivalent means in case of the therapeutic activity, either the pharmacological activity or the clinical activity, clinical effect of the drug must be same. That is a dynamic property of the drug must be same. So usually the dynamic properties may vary based upon the stereochemical nature of the drug molecule. Sometimes rest all the characteristics will be same, but based upon the enantiomers it can vary. So the therapeutic equivalence ensures the equality in the pharmacodynamics. Okay, though this is the equivalence definition, let it be this you have already gone through. Pharmaceutical equivalence, therapeutic equivalence and clinical equivalence, this you can read when you get the time. Okay, next one is in vitro bioequivalence. Bioequivalence can be performed in two ways. The term bioequivalence means purely in vivo, but bioequivalence can be predicted in vitro and in vivo bioequivalence also. Okay, so what is the need of bioequivalence? When a generic company make a product equal to that of the innovator, we perform the bioequivalence. Apart from that, various other situations also we need to prove both the drug products are same. Suppose for example, I am a generic company, I am having my own drug product, but I have changed one of my excipient. I found a scarcity of an excipient, I found some other issue with excipient. Particularly sometimes the excipient available from the manufacturer as the raw material as itself has changed. Some by means of some situation we may have to re-perform the bioequivalence studies. In that situation, some studies we can restrict only with in vitro data, and some studies we have to go for in vivo data itself. What are the conditions we can restrict only with the in vitro data, and what are the conditions we must go for in vivo bioequivalence? In vitro, the first one is the drug product differs only in strength of the active substance. Suppose the drug product is having 50, 100, 120 mgs of drug, and I'm need to perform the bioequivalence of all the strength. I hope you got the point. The particular drug molecule, if you take atorvastatin available in 5 mg, 10 mg, 20 mg, 40 mg, 80 mg, no need to perform the bioequivalence of all the strength. You have to perform the bioequivalence of only one strength. Remaining all the strength you need to prove only by in vitro method. Hope the point is clear. Out of 10, 20, 40, 80, 120 mg, you need to perform only the bioequivalence with 80 mg. Remaining 10, 20, 40 and 120 you have to do only with in vitro equivalence test. That's the thing. Second, the drug product has been slightly reformulated or manufacturing method has been slightly modified. Okay, reformulation, making a different type of formulation, manufacturing process has changed. No need to go for in vivo, but you have to inform the variation what you have made to your regulatory authorities. You have to inform whoever the regulatory authority may, because you are changing the formula or the process. So you have to inform with the in vitro data, no need of in vivo data. And the last situation is an acceptable IVIVC and an in vitro dissolution rate of new drug product is equivalent with that of already approved medicinal product. Okay, we need to prove here an IVIVC, no need to go for a clinical or a bioequivalence data. That's the conditions where we can perform the in vitro bioequivalence studies. Now the in vivo bioequivalence studies: oral immediate release products, non-oral immediate release product, modified release products. Suppose the drug is available as an extended release formulation, but the same dose or a different dose, whatever it may be, you are making it as IR tablet. The purpose of the tablet is modified. The activity of the tablet is modified. Extended release, you made it to an immediate release, or immediate release, you modified as extended release. In these situations, you have to perform an in vivo bioequivalence studies, and non-oral, like apart from you are changing the route of administration. Dose might be same, it might be pharmaceutically or a bio or a chemically equivalent, but you are changing the route of administration. There you have to perform the in vivo bioequivalence studies. Okay, now what is the experimental design for in vivo bioequivalence design? So 4 different experimental designs we are selecting. One is completely randomized design, randomized block design, repeated measures crossover and carryover design, Latin square design. What is meant by completely randomized design? So we need to compare a test with a standard. The selection of the study design is based upon the individuals, or based upon the volunteers on whom we are going to conduct the study, based upon the population. I think you may be aware about this type of studies. In completely randomized design, for conducting the bioequivalence study, randomly we are selecting the members. We are not setting any of the criteria for selection of members, and no criteria for administration of drug to the individual. You have 30 members in your class, you have boys and girls, you people are of almost same age, but the body conditions are different, body physiological conditions are different, BMI is different, you individually all are very. So in case of a completely randomized design, to perform the study, randomly I am selecting 20 members from you. I didn't go through any of the condition, and from this 20 members I made 2 groups, 10 members first group and the 10 members 2 group, no criteria for division. And first to any one group I given the reference product, other group I give the test product. So the selection of the volunteers, administration of the drug, everything is randomized. There is no criteria. This is the randomized, completely randomized design. Now the second one is the randomized block design. So in case of this randomized block design, what you have to do is selection of people will be homogeneous. Means male means only the male. Studies are conducted only in males. Female means only in females. A specific BMI, a specific age, specific disease conditions, or medical background, medical history. So by maintaining some inclusion and exclusion criteria, the population we made into a uniform. The population where we are going to test our drug product will be uniform. Okay, and this uniform people, I am selecting 20 members. I made into 2 blocks. First block, second block. So both the blocks are having the people with the same characteristics. First block I will give my reference product. Second block I am giving my test product. So this type of study is also known as a parallel study, because for the reference and the test product, the study is conducting at the same time in a single time period, and the reference product is giving only for the first group, and the test group is giving for the tested drug product is giving for the second group. This is a parallel design. In case of repeated measures, crossover and carryover design, this is a crossover design. So here is an example of a crossover and a parallel design. In the crossover design, the first group, the selection of group will be homogeneous based on inclusion and exclusion criteria. We may divide the people into, if we have two product to test, a reference and test, we divide into two. First group will administer in the first study period, will give the test. Second group will give the reference. In the second study period, the group who received the test initially may receive the reference in the second time period, and the initially who got the reference may get the test in the second time period. Now all the members got both the test as well as reference. This is a crossover design. Who got test first, they will get a reference in the second time period. Who got reference first, they will get a test in the second time period. In between these two, a washout period is given in order to eliminate the entire amount of drug from the body. You know, minimum 7 to 9 half-lives are required for a drug molecule to eliminate from the body. 7 to 9 half-life. So based upon the half-life of the drug molecule, the washout period can be decided. Some molecule may be 7 days only. Some molecule it may extend up to 1 month or 2 months. So after the first time period, we will provide a washout period. After the washout period, we can go for a second study. This is what repeated measures, crossover, carryover study designs. Okay, the last, so this is the most advantage one. The reason is, number of people are getting both test and reference. This people what we have selected is homogeneous. Everybody is tested. Number of individuals are high. The chances of getting the better results are high in this case of crossover designs. The last one is the Latin square design. Latin square design is also a type of crossover design. Only difference in Latin square data is the division of groups. Suppose you have 3 drug product to be tested, 1 reference and 2 tests. 1 reference and 2 tests, totally 3 drug product to be tested. In that case, you can divide the selected population in 3 groups. First group, first a reference, second group first test, third group second test. And in second time period they will interchange, third time period again interchange. So 3 formulation means 3 group, 3 time period. This is a square in shape, 3 rows, 3 columns. In the rows they can mention the study period, in the columns they can mention the subject number. If there are 5 product to be tested, you can divide the entire population into 5 group, 5 study periods, 5 formulation. This is a Latin square crossover design. Now the last part is bioequivalence study protocol. I think you might have come across this, even though we'll explain this. Protocol, as you know, protocol is a written document that is made by the industrial people, and it is a written document made by the industrial people and hand over to the clinical trial institutions or clinical research organization regarding the method for conductance of clinical trials, which is highlighting the protocol may highlighting the details regarding the product, details regarding the inventor, the mode of administration, what is the drug product it is, selection of population, what are the criterias, etc. So what are the details that must be included in a clinical trial protocol or a bioequivalence study protocol is mentioned here. So we have around 10 points here. I'll just explain one by one. Title, who is the principal investigator, what is the project number and date, what is the objective of the study, what is the study design, crossover, Latin square, what is the design might be, what are the drug product, which is the test product, what is the reference product, ഇതിനെല്ലാം details you have to submit. Dosage regimen, what is the frequency of administration, TID, BID, or sample collection schedule, at what interval the sample must be collected, what interval the sample must be collected, which blood samples must be collected, housing, whether the patient must be under care or can be sent off, or what is the food habit must be taken care by the patient, by the individuals, fasting or meals schedule, whether the drug must be given in a fasting or after the meal, and what type of meal must be administered, because this type of drug molecules, the food habit can vary the rate of absorption. The food habits can affect the rate of absorption. So the particular drug molecule, what all food can be given for the patient. The individuals are not allowed to take the type of food what they wish. If that varies, means surely there will be variation in the absorption or the kinetics of the drug molecule. So all the people must have a same type of food, means especially the oil content, the protein content, etc. Varies can affect the kinetic parameters. Analytical method, so after collecting the blood sample, what analytical method you have to opt to determine the plasma concentration. Study population, which type of subjects, how many subjects, what should be the medical history, what are the physical examination must be conducted, what are the laboratory tests, because some patients with a different medical background may affect, or on medication can affect some of the interaction with the study what you are going to conduct. So the exact medical history must be properly prescribed, and the physical examination of the patient also must be conducted, and the laboratory test must be conducted, and that also must be within the range. The same we select given some exclusion. In the next point is inclusion and exclusion criteria. Same like inclusion and exclusion criteria, the laboratory test parameters also must be within the range. Some people may be having some habitual effect, like some cholesterol level is high, some people with diabetes, so those people should not be selected. They can prescribe at what characteristics of what should be the level, at what level of hemoglobin the patient must be selected, at what level of the liver enzymes the patient must be selected. So the laboratory test results also they can prescribe here. Now restrictions and prohibitions, if the people who are undergoing the study, if they have any restriction, that must be mentioned. Clinical procedures like dosage and dose administration, biological sampling schedule, activity of the subjects you have already seen before. Ethical considerations, what are the basic principles in ethics, institutional review board, who are the review board members, and informed consent, what are the matters should be printed on the informed consent, preparation of informed consent, etc. And indications for subject withdrawal, at what conditions, or what toxicity conditions, or what side effect conditions, what ADR if observed, the patient must be withdrawn. Then adverse reactions and emergency precautions, what must be given, what are the minimum facilities required to conduct the studies, data analysis, how to perform, means data plasma concentration you got, and what are the method used for data analysis, analytical validation procedures, how to validate the analytical procedures, then statistical fixing of the data. After getting the data, how to compare, which statistical method is required used to compare the data, and accountability of the drug molecule what you are giving, how to preserve, and what account section must be given, and in appendix what are the any other relevant data witness ticket can be included in the appendix. So these are the details must be added as an bioequivalence study protocol. And last one is the statistical interpretation. Here you have the statistical treatment of data. Generally the statistical interpretation is based on ANOVA as well as based on confidence interval approach. We can compare the test as well as the reference product. That's okay. So with this your bioavailability and bioequivalence chapter is completed. Okay, so you have come across bioavailability, determination of bioavailability, in vitro dissolution method, acceptance criteria, stages of dissolution, what is in vitro in vivo correlation, bioequivalence study, how to conduct the bioequivalence study, what are the bioequivalence study protocols. Okay, so learn this chapter, and any doubts you can clarify with me. Okay, thank you.